right. Good morning, everybody. Here is our remainder of our slides from this week. Hopefully, this is going to be helpful. If you have any questions on this, just post it in the comments underneath the video. And hopefully, I can get back to them and you guys can get some good studying in this weekend. So, let's get started. Where we left off yesterday was talking about the Casparian strip. So, talking about how anything that needs to get into the xylem has to get past this Casparian strip, and it has to go through the cytoplasm of the endodermis, so through that symplastic root. Now, one of the results of this movement of water is this thing called root pressure, and this is basically an upward pull of the xylem sap, and it's a pressure that occurs in the cells as the water is moving from high water potential to low water potential. So it can actually produce this really interesting thing, and I'm sure you guys have seen this at some point when you've watched dew on the water, dew on grass, anything like that. But this is a really good example of it. I thought this was an amazing image. And it's called guttation. So guttation occurs when root pressure causes more water to enter the leaves than is transpired. And so this is basically saying, okay, water is always going to move from an area of high water potential to an area of low water potential. But this doesn't also always mean that the leaves are capable of holding all of the water that wants to move into their cells. So what ends up happening is that can actually be forcibly pushed out of the cells to create these little bubbles. It's a fascinating process. So when next time you guys look at blades of grass in the morning, it may not be dew on them, but it may be the force of root pressure. All right, quick reminder of some properties of water. So one of the things that we talked about very early on in the beginning of the year were these properties of water called cohesion and adhesion. So this is going to be, cohesion is water sticking to itself, and adhesion is water sticking to another surface. So in this case, we're looking at the surface of the cell wall. And these two things, the reason that we're reviewing these is because cohesion and adhesion are essential to the movement of water in the xylem. And this image is a really good sort of summary of what's going on here. All right. So over here, you guys, we have the roots, and then we have a bit of the trunk, and then we have the leaves. And this is showing how xylem pressure and xylem transport is going to occur because of cohesion and adhesion. So if we start here where water is being water is getting into the roots, and as it flows upwards through the xylem, it is going to only be able to do this because of cohesion and adhesion. So as the water enters the roots and starts traveling through the symplast, the cohesion and adhesion, so the cohesion is going to be water molecules that want to attach to themselves because they're attracted to each other. Remember, those hydrogen bonds are attracted to the oxygen bonds in water, so they're going to be attracted to each other. And adhesion means they are going to be attracted to the surface of the xylem cells. And so this is literally going to pull the water upward through the roots because of these attractive forces. Now, if you don't remember these forces of cohesion and adhesion, I encourage you to go back and look at your notes and review those properties of water. But this property of cohesion and adhesion is going to be how water gets all the way to the leaves of the tree and then eventually leaves via the stomata. Now, one thing we haven't talked about in this image yet is if you'll take a look at this region over here with the red box. So this is going to be the water potential gradient starting from the roots and ending in the leaves. So let's take a look. As we get to the root water potential, again, it's measured in megapascals, not in bars in this case, but the soil water potential, so this is not the roots yet, but this is the soil water potential, has a water potential of negative 0.3 megapascals, which is less negative than the trunk xylem. So it means it's more positive. It means it has a greater water potential than the xylem. Where does water move? Always from an area of high water potential to an area of low water potential. So if we take a look at the roots, the area around the roots is an area of high water potential. That's all this blue stuff here is water. So that high water potential is going to move into the roots based exactly on the process that we observed in the lab with the potato cells. This water potential gradient is going to apply throughout the entire anatomy of the tree. So it's going to, starting from the soil and the roots, the water potential is going to decrease as you get farther up the tree. And this decrease is going to pull the water upwards in the tree. So if you can see the trunk xylem compared to the trunk xylem down here, so the trunk xylem a little bit farther up in the tree has a water potential of negative 8 megapascals. So it's going to have a significantly lower water potential than the roots, and it's going to pull water upward. Same with the cell walls in the leaf. It's going to have a more negative water potential than the trunk xylem, as well as the leaf air spaces. So these are going to be the spaces around the mesophyll cells in the leaf. It's going to pull the water into the outside air, which has a water potential of negative 100 megapascals. So you can see, based on our concept of water potential, how this pulls water from the roots to the tips of the leaves. But why is the water potential so much lower at the leaves than at the roots? Now, this is going to be a little bit more complicated, so buckle up and get ready for some fun water potential stuff. This is because of transpirational pull. So, when we are looking at if we are looking at the spaces outside of these mesophyll cells, and I know I'm skipping around a lot, but I'm just hoping that you guys can pause this whenever you want and kind of take a break and process what I'm saying and then move on. So feel free to do that. But the transpirational pull is saying before water molecules evaporate from the stomata, they coat the surface of the mesophyll spaces and form crescent-shaped menisci. So let's go back to that image that we had of the different layers of mesophyll cells. So we had the spongy mesophyll cells and the palisade mesophyll cells. The spongy mesophyll cells had a bunch of space around them where air could mix in. Now, these mesophyll cells are also going to be surrounded by water. They are going to have essentially what you can call a meniscus, a layer of water, a film of water around those spongy mesophyll cells. Now, depending on the water potential of the air around those mesophyll cells, the curvature of the film of water is going to change. So if the area around the mesophyll cells has a very low water potential, it is going to have a stronger pull on the water, on the film around the mesophyll cells. And that stronger pull is going to increase the pressure on the water around the cell. So the curvature of the film of water, so that's talking about this, how curved it is going to determine the pressure on the water. So the greater the curve in this case, and the arrow is showing the amount of pressure pulling water out, but the greater the curve, the higher the pressure. The higher the pressure, the higher the water potential. Okay, so if that was confusing to you, make sure you go back and look at our water potential equation. So the higher the water potential, the higher the pressure, the higher the water potential, all right? And that's going to cause more water to transpire out of the cells and out of the film around those mesophyll cells. As water molecules evaporate from the stomata, the curvature of the film of water inside the leaves increases, and the water tension increases. So water tension is going to be saying the same thing as the water pressure. It's basically, they're sort of synonymous in this case. And this is going to be due to the cohesiveness of the water molecules. So remember, cohesion is water molecules attracted to each other. They want to stay together. So in this case, the water molecules are going to want to be attracted to each other. And so as the curvature of the water around those mesophyll cells increases and becomes more aggressive, it's going to cause more water to evapotranspire and to transpire out of those stomata and out of those leaf air spaces. Okay? So, meniscus tension, and I wanted to bring this up because it's a statement in your book that I felt like was important. And I realize there's a lot of text in these slides as well, but hopefully the text is helpful to you in sort of translating what the book is trying to talk about. So, meniscus tension, so the tension due to the curvature of this film of water around the mesophyll cells, meniscus tension, is inversely proportional. So inversely proportional means if it increases, then the other thing decreases, to the radius of the curved surface. So meniscus tension increases, the radius of the curved surface has decreased. Meniscus tension decreases, the radius of the curved surface has increased. As the meniscus becomes more concave, so this is going to be an example of a more concave meniscus, its radius decreases and the tension increases. Okay? This is an important statement for you to understand. I wanted to throw it in here because you guys need to practice using your AP terms. So inversely proportional is going to mean one thing increases and one thing decreases. Now, there was a more mathematical explanation for that, but that's a better opportunity to use with a whiteboard. All right, so this tension, the tension that we were just talking about here, about the meniscus and the radius of the curved surface, the tension produced in that meniscus pulls water from the areas of greater pressure, so the fluid-filled areas like the xylem, into the areas of lower pressure like the stomata. So areas like the xylem are going to be experiencing a high level of pressure due to cohesive and adhesive forces that are pulling water upward through the xylem. This is going to cause the water to move into an area of lower pressure in the region of the air spaces and out of the stomata. Okay? All right, again, please feel free to pause. I hope you guys are studying with your friends. Make sure to post your pictures with your friends when you are studying with them this weekend, and you'll get extra credit. But this would be a good opportunity to pause and sort of ask each other questions and make sure that you're catching up and kind of tracking with what we're talking about. All right, as I said before, we were going to go over guard cells in more detail. So the guard cells of the stomata are going to be, there is going to be a little bit more detail that you guys need to know for the stomata. So, as we know, guard cells are going to open and close the stoma. So, a single stomata is a stoma. They're going to open and close by changing shape. Now, as I've said, swelling, more water, swelling of the guard cells means an open stomata. And this is because of these radially oriented cellulose microfibrils. So if we look at these radially oriented cellulose microfibrils, this is going to be these lines in the guard cells here. they're going to be closer together. Think about them as strings on like a balloon or something like that. These are going to be closer together on the inside of the stomata than they are on the outside of the stomata. So that's what this red image, this red half circle is showing here. The radially oriented cellulose microfibrils constrict the movement of that side of the cell. So as water fills the guard cells, though that region of tightly constricted cellulose microfibrils is going to decrease the flexibility of those cells, while the outside of those guard cells has more flexibility, and so they can extend more than the inside can when they are being filled with water. So this results in an uneven swelling of the cells. And so when the water releases and when the stomata need to contract, these cellulose microfibrils don't have as much of an impact on the shape of the cell, and so they're going to close the cell because they are not full with water. And so the radial orientation of the cellulose microfibrils isn't going to have as much of an impact on a closed stomata. So, one thing, oh no, this is the right image. Cool. So, one thing that we need to notice about this image, now ignore this top purple box right here for a second and just take a look at the image, because we've seen this image before. We just need to go through it in a little bit more detail. What we're seeing in this image is the movement of water as well as the presence of solutes. So in this case, the solute we're looking at is potassium ions. So what a plant can actually do at some points is, in relation to the potassium ions surrounding the guard cells, the guard cells can take up potassium, so they can increase the transport of potassium across the cell membrane and increase the solute concentration inside the guard cells. Now, increasing the concentration of the solute, increasing your solute potential, means you are increasing your water potential. Yes. And so water is going to move into these cells. Okay, water is going to move because think about it as about your hypertonic and hypotonic. Water is going to move from an area of high concentration to an area of low concentration. The potassium has decreased the solute potential. Yeah, that's it. That's why I got it backwards. Decrease the solute potential, not the concentration. Decrease the solute potential, so it's going to decrease the water potential. So water potential is going to go from an area of high to an area of low water potential, and so water is going to rush into the stomata to an area of lower water potential. So just to clarify, because I said it wrong originally, just to clarify, potassium is going to increase inside the guard cells. An increase in potassium decreases the solute potential of the inside of the guard cell. The decrease in solute potential decreases the overall water potential of the guard cells. So water is going to go from an area of high water potential, which is outside the guard cells, to an area of low water potential, which is inside the guard cells. Similarly, if a stomata needs to close, that it can actually release a lot of its potassium into the surrounding cells. The release of potassium is going to decrease the solute potential. Decrease the water potential. So water is going to move from an area of high water potential to an area of low water potential outside of the cell. Sorry, I kind of screwed that up a little bit, but hopefully you get it. Hopefully you get the picture. All right, and this goes into a concept that you guys are going to see all the time. you need to be very well aware of this concept of biological trade-offs. Now, this is going to come into play most of the time when we're talking about behaviors and adaptations, but this is a really good example. You will definitely see a question on biological trade-offs somewhere on your AP exam. So this is going to be when one behavior or adaptation, so in this case, we're talking about the stomata. The stomata would be the behavior or adaptation. And the benefit of one behavior, so closing the stomata, comes with an equal cost to the organism. So it's a cost to benefit ratio that we are looking at in these biological trade-offs. So in this example, closing the stomata is going to result in a lack of CO2 for photosynthesis. So the plant is not going to be able to add any more sugars to its mass. It's not going to be able to grow because its stomata are closed. Now, opening the stomata also means losing water that the plant cannot afford. So this is in a situation where it's an extremely hot day, the plant is becoming dehydrated, and so it might close its stomata. But closing the stomata means it doesn't have any CO2 for photosynthesis, and so the plant is going to stop producing sugars. So this is the concept of a biological trade-off. If you remember one example of a biological trade-off, this could be an example. There are a lot of examples, but this is one that we've talked about. All right, last concept, sugar transport in the phloem. So this is going to be 36.5, and we haven't really talked about the phloem yet. So this is going to be where we start to talk about the phloem a little bit more. So translocation is going to be the transport of sugars to the phloem by pressure flow from what is called the source to the sink. So this is transport of sugars into the phloem from source to sink. So the source is going to be the region of the plant that is producing sugar, is doing photosynthesis. So this is going to be your leaf cells, your mesophyll cells that are producing sugar for the plant. And the sink is going to be an area that needs to consume or store sugar. So this is likely going to be like a root cell or another type of cell that isn't producing sugars. It's not doing photosynthesis, but it might be doing other things. It's going to be doing xylem transport. It can be doing support cells. It can be a lot of other things. So that's going to be translocation, the movement of sugars from source to sink. And this loading of sugars from source to sink occurs in the sieve tube elements or members in the phloem cells. So these are basically the vascular cylinders of the phloem cells. It's the sieve tube element. So that's this cell right here in this image. It's the one that the purple arrow is pointing to. These are going to be our phloem cells. And so what ends up happening is if we take a look at this image, we've got our mesophyll cells, we've got our apoplastic root, so that's this blue line here, and we have our symplastic root. Same as xylem and phloem. We have an apoplastic root and a symplastic root. So in the symplastic root, sugars can easily be transported in the mesophyll cells where photosynthesis is occurring through the symplast and into these sieve tube elements or sieve tube members. They're the same thing, but they're just, for some reason, the book calls them sieve tube members, but the scientific community mostly calls them sieve tube elements. So these sieve tube members, they don't actually, we talked about this a while ago, but the sieve tube members don't actually have their own nuclei. They don't have their own cell organelles. They don't have a lot of the organelles that most cells would have because they're focused on the transport of sugars. So what they end up having is a companion cell. And this companion cell is going to make up for those functions that the sieve tube member does not have. So that's this companion cell right here. This is the sieve tube member, and this is the companion or the transfer cell. And this companion and transfer cell is going to be how a lot of the sugars and organic molecules get into the sieve tube member. And that's what this is talking about here. So in order for sugars to be transported between sieve tube members, they have to go through this sieve plate. And that's how they get transported through the sieve tube element. And we're going to talk a little bit more about how molecules and sugars move through the sieve tube elements. All right, let's see how many more we got left. We got 3 left. Almost done. So the loading off of sucrose into the phloem, it actually occurs via active transport. So go back to our membrane transport systems. This is a lot of good review. Active transport is going to require ATP. So this sucrose loading is going to occur via a proton pump. So we talked about a proton pump a couple lectures ago. Protons are going to be pumped from an area of low concentration to an area of high concentration. This requires a lot of energy, and so ATP is going to have to be inputted into this system to make this happen, to keep this high concentration of protons over here. The high concentration of protons is going to want to move down its concentration gradient. So it's going to move down its concentration gradient from an area of high proton concentration to an area of low proton concentration. This desire to move from an area of high concentration to an area of low concentration is going to produce a lot of potentials for this cell. We talked about this at the beginning of this unit. And this can use a co-transporter. So sucrose, if it needs to get inside the cell, it can use this proton gradient and use a co-transporter, so something that requires both a proton gradient as well as the sucrose molecule, to then transport sucrose inside the cell. So this mechanism is going to be how sucrose, if we go back to this image, this is going to be how sucrose can enter the sieve tube member, is by this process of co-transport and active transport. Now, that was this image over here. So the transport of sucrose across the membrane, it is only going to be able to occur via co-transport because sucrose is too big of a molecule to just diffuse through the membrane or to go through a protein channel. It's going to have to require a very specific co-transporter in order to get inside the cell. So if we're looking at this image, now this image is on page 753 in your textbook. If you want to take a look at that, you can take a look at the whole image. Now I've divided this into 2 parts. Now what you can do before you look at this slide, if you go to the image on page 753, I would recommend, as we do in class very frequently, that if you're studying with a group or even if you're studying on your own, try to explain and in your own words, literally type out or write out, whatever works for you, explain figure 36.18. So explain the transport of sugars from source to sink in this image. All right? So let's go to this image. Go ahead and pause it if you're going to try to explain it, but I'm going to explain it for you guys right now. This should be the last thing that we go over. So translocation, movement of sugars from a source to a sink, is going to occur in this manner. You've got a sucrose source here, so this is going to be a mesophyll cell where photosynthesis is occurring and sucrose is being formed. Sucrose can then be transported into the sieve tube element via this active transport mechanism that we just talked about. As it's transported, it's going to very dramatically increase the amount of solutes in this sieve tube element. And as we've talked about, increasing the amount of solutes decreases the solute potential of the region. Decreasing the solute potential is also going to decrease the water potential. This decrease in the water potential is going to cause water to rush from the xylem vessel into the sieve tube element. As the water rushes from the xylem into the phloem, because of this change in the concentration gradient of water, it's going to increase the pressure of this sieve tube element. So think about this as filling up a balloon with water. If you keep filling up a balloon with water, the pressure on the membrane of that balloon is going to increase. And that's exactly what's happening here. So the increased pressure forcibly pushes the water that is now including all of those sucrose molecules, forcibly pushes those molecules down the source to sink gradient. So more water is going to mean more pressure, forcing the sap from the tube. Now, the direction of this force is going to be based on the sieve tube elements. And so the sieve tube elements are going to determine which direction this pressure is going to go. As the pressure reduces, once the sugars are from the source to the sink and the pressure has decreased closer to the sink, so the pressure reduces and gets closer to the sink, the water potential, so the sucrose is being transported into a storage route. So in this case, the source up here, so the mesophyll cell up here, is transporting sucrose into the root cell. This transport of sucrose from the leaves to the roots is going to then increase the solute potential. You've decreased the amount of solutes, and so you're increasing the solute potential of this phloem cell. The increase in solute potential is going to increase the water potential. And then the water is going to move from an area of high water potential to an area of low water potential on this side. And that's going to be how you get this transport. Oh, I thought I had the whole image. But that's going to be how you get this transport of phloem and sucrose in the phloem from an area of high concentration down the gradient to an area of low concentration. So from an area of a source, so a mesophyll source cell, to a sink, right? So hopefully that was helpful. If you have any questions at all, please feel free to ask me. Feel free to comment down below, and good luck studying, guys.